Hello everyone. So in the yesterday class, we have discussed up to 51 questions. So we are continuing the from 52 to 100 questions. So now let's try to understand all the questions in a simple way. So now today we are going to discuss most of the questions from the dark reaction as well as some of the questions from the light reactions also. Because 90% of the cases from the photosynthesis, we can expect the question from the dark reactions only. So now listen, in a controlled experiment of photosynthesis, carbon dioxide supply is suddenly withheld. Which of the following will accumulate in the plant cell? So friends, what is the question saying that they have stopped the carbon dioxide supply all of a sudden? Then they are asking which of the following can be accumulated in the plant cell. Every student very commonly know that who will actually accept the carbon. Friends, we know very well RUBP is the one which is going to accept the carbon. So RUBP accept the carbon. So if there is a no carbon, if there is a no supply of the carbon, RUBP will get accumulated in the plant cell. Hence the correct answer is B. Now, which set of terms most accurately describe the pathway taken by a molecule of carbon dioxide from the atmosphere to the point where it enters the Calvin cycle? This question seems to be very simple question, but very logical question. If you don't understand the question very correctly, friends, you can't answer the question very correctly. Okay. Now, first, every student try to understand how does carbon dioxide will enter first of all. That is the very important point for the exam. You know very well all the gases can enter into the plant via what, friends? Stomata. So obviously true. And you also know very well, I have explained it to all of you, where the stomata is present, friends, in the lower epidermis, which is I mean to say to the lower epidermis, which cells are very close, mesophyll cells are very close. In the yesterday class also I have explained you, and I also explained you that mesophyll cells are having how many I mean to say it is having large intercellular spaces, because friends all the mesophyll cells are associated with each other with a lot of gap. Correct, na? So what is the answer? Stomatal aperture, intracellular space, mesophyll, chloroplast. Hence the correct answer will be the number 1. Now, carbon dioxide joins the photosynthetic pathway in. What do you mean by that? So at which reaction the carbon dioxide will enter into the photosynthetic pathway? We all people know very well, we are going to reduce the carbon into glucose. In which reactions, friends? Undoubtedly in the dark reactions only. Hence, what will be the answer? The answer will be the 4. All the students now listen to me carefully. Light energy is converted into chemical energy during a light dependent phase, and chemical energy is used to. See friends, we studied very clearly what is the ultimate goal of converting the light energy into chemical energy. By using that particular chemical energy only, the plants are going to reduce the carbon into glucose. For this particular process, what is not required? Light is not required. So reduce carbon dioxide to the sugar, nothing but carbohydrate, during light independent phase. Hence friends, what will be the answer? Undoubtedly answer is the third one. During photosynthesis, PGA is changed into phosphoglyceraldehyde. Which of the following reaction occur? So we know very well, PGA is how many carbon? 3 carbon. Correct, na? And glyceraldehyde 3-phosphate, it is also a 3 carbon. Then what happened, friends? One form is reduced to the another form. So PGA converted into GA3P during the step of the reduction. Not to get confused with that. Now the correct answer is what? Number 2. That is the reduction. So question number 57 says that ribulose bisphosphate carboxylase enzyme catalyze the reaction between. So they are asking what is the first step in the Calvin cycle. We all people commonly know that what is the first step in the Calvin cycle? RUBP combined with the carbon dioxide, and it is going to give rise to what? It is going to give rise to 2 molecules of 3-PGA. And friends, this is the reaction which is catalyzed by the Rubisco enzyme. Is it? Hence, what will be the answer? Carbon dioxide and ribulose 1,5 bisphosphate. The correct answer is the option 2. Think and answer. During Calvin cycle pathway, ATP and NADH is utilized for the reduction of, very good question it is. See, you know, right? In the first step is what, friends? First step is the fixation. Yes or no? Of course, yes. See, in the Calvin cycle, there are three steps are there. One is called fixation. Second one is called reduction. And friends, third one is called regeneration. Every student commonly know that in which steps we actually spend abundant amount of energy. We spend energy in the reduction also, in the regeneration also. Anyway, they did not ask the question anything about regeneration. They asked the question only reduction. So what we reduce? We reduce the PGA. Hence the correct answer is PGA. Now, this is also a very interesting question. In terms of the spatial organization of photosynthesis within the chloroplast, what is the advantage of light reaction producing ATP and NADPH on the stromal side of the thylakoid membrane? See, friends, the question is very valid question. What they are asking, in the light reaction, we produce ATP, we produce NADPH2. That is true point. They are asking the question, why the plant will produce ATP and NADPH only on the stromal side of the thylakoid membrane? What is the reason? Because we all people know that on the stromal side of the enzyme only all the enzymes are present. Stromal side of the thylakoid membrane, rubisco, pep carboxylase. So they will do the reduction reaction. For the reduction reaction, friends, they have to compulsory use the energy. So what is the answer? The Calvin cycle which consumes ATP and NADPH occurs in the stroma. That's why, friends, ATP and NADPH are synthesized on the stromal side of the thylakoid membrane. Right. Now the question says that how many ATP is needed to generate one molecule of RUBP in the Calvin cycle? And today morning class also I have asked you the same question. I hope every one of you remember. So in order to regenerate one RUBP, we require one ATP. Actually, friends, how many RUBP we can regenerate? You remember, we regenerate 3 RUBP. So friends, 3 RUBP, we require 3 ATP. But in the question, they have asked only 1. Hence, the answer will be only 1 ATP. Not to get confused. And see here, this question also, most of the students are giving the wrong answer. The scientist believed that since the first product is the C3 acid, in the C3 plant, what is the stable product? PGA. PGA is how many carbon? 3 carbon. So what is the question they asked? What they believed. It is not asking the true situation. Please don't get confused. They are not asking about the true situation, what they believed. So intermediate is 3 carbon. They thought that whichever is accepting the carbon, that will become 2 carbon. So 2 carbon plus 1 carbon will be accepted and it become 3 carbon intermediate. So that is what they believed. So answer is what? C2. Most of the students are giving the answer as a C5 or C6. No, they are asking if intermediate is 3 carbon, what is the starting compound will be? What they believed? They believed it's a 2 carbon. Not to get confused with that. Okay. Select, once again, try to understand. They're asking incorrect. Select an incorrect statement for biosynthetic phase of photosynthesis. Biosynthetic phase means, always remember, friends, synthetic phase or the reduction phase, anything is what? Dark reactions only. Option 1 says that, and they're asking incorrect statement. This process does not directly dependent on the presence of light. Of course, true statement. This process is dependent on the products of light reaction. Of course, true. What are those products? We know very well. It is dependent on the ATP. It is dependent on the NADPH. Option 3, immediately after light becomes unavailable, the biosynthetic process continues for some times and then stop. Of course, true statement. All the students listen my words correctly. Whenever light become unavailable, even in the absence of light also, the process will be continued until they prepare their food. Once the food is prepared, then obviously the process is going to stop. What will be the fourth one? Use of the radioactive N15 in algal photosynthesis studies led to the discovery. Absolutely wrong statement. We did not study N15 in the algal photosynthesis at all. Hence, the incorrect statement is what? Option 4. Most of the students will think that option 3 is the incorrect one. Think and answer the questions. Now, which of the following statements regarding C4 plant is a false. They're asking false. The first statement says that the primary carbon dioxide acceptor is a 5-carbon molecule. This is only wrong statement, right? Every student know that in case of a C4 plant, who is the carbon acceptor? PEP. PEP accept 1 carbon and it is going to give rise to what? It is going to give rise to the oxaloacetic acid. So PEP is how many carbon? It's a 3 carbon. But what they are saying in the statement, the primary carbon dioxide acceptor is the 5-carbon molecule, which is the wrong statement. Option 2, the initial carboxylation reaction occur in the mesophyll. Of course, true, right? And Calvin cycle does not take place in the mesophyll. Of course, true. Because friends, in the C4 plants, we studied very clearly where the rubisco is present. Rubisco is present in the bundle sheath. Hence, this is also true statement. Leaves that fix carbon dioxide has 2 cell types. Of course, yes, mesophyll cell and bundle sheath. This is also true statement. What is the wrong statement? The number 1 statement. Easy to answer. Now, question number 64. In C4 plant, bundle sheath shows. In today morning class only, I have explained it to all the students. Mesophyll having a large intercellular spaces. Yes or no? And they present very close to the leaf surface. What did I told to all of you about the bundle sheath cell? No intercellular spaces. Correct, na? And they are very, very, very thick walled. So what will be the direct answer? It is no need to think also. They are thick walled cells having many chloroplast and no intercellular spaces. Don't get confused with this. Hence the correct answer is what? Option 3. Now, question number 65. This is also very important question, friends. Never ever forget this. During the synthesis of 1 molecule of glucose, as a net gain, the number of carbon dioxide molecule required and turns of Calvin cycle to be operated respectively. In the morning class also, I have explained it to every student very clearly. Always remember, for half glucose and 1 glucose, it becomes easy for all of you. To produce a half glucose, we require 3 carbon dioxide. So the cycle has to turn 3 times. So 3 Kelvin cycle. So friends, they are asking for 1 glucose. So to produce 1 glucose, we require 6 carbon dioxide and we require 6 Kelvin cycle. Yes or no? So then what is the answer? 6 and 6. Not to get confused with that point. Next one, which does not show HSK pathway. You remember what I told you? C4 pathway can be shown by more than 18,000 type of the angiosperm. But friends, I mean to say monocots. But I am giving you some examples of exceptions. Sunflower, amaranthus. They, see, amaranthus is very special, friends. What is the specialty of amaranthus, you know? Amaranthus is dicot. Remember my words. Amaranthus is dicot. But since even though it is a dicot, it can follow the C4 pathway. This is a very important point. Sunflower originally cannot show the C4 pathway. Why? Because impaired cranch anatomy. I want you to remember the reason also. Because of the impaired cranch anatomy or the irregular development of the cranch anatomy, the sunflower do not show the HSK pathway. Hence, the correct answer is 4. And very important question. Always this kind of questions are more important for the exam. Total energy requirement for fixing 1 carbon dioxide in a C4 cycle. In the morning class, I have explained you many times to all the students. In order to fix 1 glucose, how many carbon dioxide are required? I'm talking about 1 glucose. 1 glucose, we require 6 carbon dioxide, 12 NADPH, and how many ATP we required? We require for 30 ATP. So friends, listen. This is the calculation for 1 glucose. But in the question, they did not ask for 1 glucose. They are asked for 1 carbon dioxide, how many NADH and how many ATP will be used. So if 6 carbon dioxide, 1 NADH means for 1 carbon dioxide, 2 NADPH, easy question. If 6 carbon dioxide, 30 ATP means 1 carbon dioxide, friends, undoubtedly it is the 5 ATP. Is yes or no? Hence the correct answer is the 5. Very, very important. because calculation questions are always important for the exam. Number of carboxylations in the C4 cycle. This is also very, very important question. What do you mean by the word carboxylation cycle? Where there is a addition of the carbon, not to get confused with that. In the C4 cycle, obviously we studied many number of times. There are two carboxylation cycles will be taking place in the C4 cycle. Direct question it is. C4 plants are more expensive in terms of ATP required for carbon dioxide fixation. The additional number of ATP required by C4 plants in comparison to the C3 plant. Friends, this is the most direct question. In case of C3 plant, you tell me how many ATP we are using total? 18 ATP we are using for one glucose molecule. We studied very clearly in the C4 plant how many number of ATP we are using? 30 ATP we are using. Yes or no? Of course, yes. So how many extra ATP we used? We used an extra 12 ATP molecule. Simple point it is. Hence the correct answer is B. Very easy. All C4 plants shows cranch anatomy, and it is very important, friends. It's a very good question. Some students will think that C4 plants develop the cranch anatomy in the middle of the life. Friends, that is absolutely wrong statement. So it never be developed in the middle of the life. It is genetically determined. Very important question it is. So obviously answer is 4. So the order of 3 steps of the Calvin cycle, very important. What we discussed, first fixation occur, then reduction occur, then regeneration occur. The fixation you can also call it as what? Not to get confused. The fixation only you can also call it as carboxylation. Yes or no? Because to the RUBP, what we are adding the carbon. So adding the carbon is called as what? Carboxylation only, right? Hence the correct answer is A. Carboxylation, reduction, regeneration. For the formation of one molecule of glucose, how many turns of fixation are required? I already told you, in order to fix one glucose molecule, 6 Calvin cycles are required. Hence the correct answer is C. Which of the following statement is incorrect? They are asking incorrect. Rubisco is the bifunctional enzyme. Yes or no? Because Rubisco can fix the carbon dioxide also. Rubisco can fix the oxygen also. That's why we called bifunctional enzyme, right statement. In C4 plant, the site of Rubisco activity is mesophyll. That is wrong. See, we studied very clearly, friends. In the C4 plant, we have 2 cells, mesophyll cell, bundle sheath cell. In the mesophyll cell, what is present? PEP carboxylase is present. In bundle sheath cell only, what is present, friends? Rubisco is present. Yes or no? Of course, yes. So, option 2 says that in C4 plants, the site of Rubisco activity is mesophyll. That is wrong statement. Option 3, the substrate molecule for the Rubisco activity is a 5-carbon. Yes or no? RUBP combined with the carbon dioxide, it is catalyzed by the Rubisco. And RUBP is how many carbon? 5 carbon. So option 2 is wrong option. Option 3 is right option. Rubisco action require ATP and NADPH. Of course, yes. Where? In the reduction and regeneration. Yes or no? Of course, yes. So friends, what will be the correct answer for this? Option 2. Because Rubisco is present in the bundle sheath cell in the C4 plant. Hence, this is the wrong option. The synthesis of one molecule of glucose during the Calvin cycle. I already told you, right? Calvin cycle, 18 molecules of ATP, 12 molecules of NADPH, and 6 molecules of carbon dioxide. This is in which pathway? C3. C4 pathway, 6 carbon dioxide, 30 ATP, 12 NADPH. Not to get confused, friends. Easy to understand. Hence, the correct answer is option C. This is good question. Every student try to focus on this question very correctly, friends. It's a really good question. The question says that, friends, if there are 12 molecules of carbon dioxide to be fixed in the photosynthesis, how many turns of Calvin cycle and how many moles of glucose will be required? Most of the students will get confused here also. You have to understand, for one glucose, how many carbon dioxide are required? 6 carbon dioxide. Simple, right? For 2 glucose, How many carbon dioxide required? 12 carbon dioxide are required. Yes or no? To produce 1 glucose, cycle has to turn 6 times. To produce 2 glucose, cycle has to turn 12 times. Now, what is the question? They only given 12 molecules of carbon dioxide. So you only tell me 12 molecules of carbon dioxide means how many glucose we are producing? 2 glucoses we are producing. How many tons? Of course, 12 tons. Easy to remember. Easy question, but confused question. Okay. Now, next question. This is also a very good question. Match the following columns and select the correct option. Photolysis of water. Every student know very well where the photolysis of water will take place. Only on the non-cyclic photophosphorylation. Carboxylation of RUBP. The moment you heard the name of RUBP, where the RUBP is the acceptor, undoubtedly in the Calvin cycle. Synthesis of ATP only. ATP we only synthesize in the cyclic photophosphorylation. Consumption of 5 ATP for the carbon dioxide produced, which is C4 plants. I have explained you very clearly. Don't confuse with ATP calculations. Hence, the correct answer is 1. Easy question, logical question, important question. How many ATP are required for the generation of 1 molecule of RUBP? How many? 1. I told you, right? We have to total regenerate 3 RUBP. 3 RUBP, they require 3 ATP. So, friends, undoubtedly 1 RUBP required, only 1 ATP molecule. Easy to remember. Next, Hatch and Slack pathway is otherwise known as C4 cycle because it's a direct question, right? We are calling it as a C4 pathway, friends, because the stable product is the 4 carbon containing compound, which is the oxaloacetic acid. Direct question, easy to remember. The first step of carbon dioxide fixation in Hatch and Slack pathway in plants is, what is the first step? Combine the PEP with the carbon dioxide, and what we are going to give rise to? We are going to give rise to oxaloacetic acid. But friends, what is the point? Where it will occur? That is the question. It will occur in the mesophyll cells. Not to get confused with that point. So friends, listen every one of you. They say that formation of oxaloacetic acid in the bundle sheath. Wrong. Formation of oxaloacetic acid by the carboxylation of RUBP in the mesophyll. Right. Formation of PGA in the mesophyll cell. First of all, friends, we don't produce any PGA in the C4 pathway. Yes or no? Option is wrong. Formation of oxaloacetate by carboxylation of PEP in the mesophyll cell. That is also true, but that is not the oxaloacetate. One second. Okay, I think. Oh, friends, I'm so sorry. Option 2 is wrong option. Why? Because formation of oxaloacetic acid mediated by what? I'm so sorry. PEP plus carbon dioxide is going to give rise to the oxaloacetic acid. But in the option 2, I said that RUBP plus carbon dioxide. This is wrong, right? This happens in the C3. So what is the correct answer? Option 4. Correct, na? Formation of oxaloacetic acid by the PEP plus carbon dioxide. That is what, friends? Oxaloacetic acid. It will occur in which cells? Undoubtedly, mesophyll cell. Not to get confused. The answer is 4. C4 plants differ from C3 plant with respect to, this is also a very good question, right? Of course, in C3 plant 18 ATP, C4 plant 30 carbon. In the C3 plant, first product is PGA. In C4 plant, oxaloacetic acid. Here, RUBP is the carbon acceptor. Here, PEP is the carbon acceptor. So, friends, all are differences only. Correct, na? Hence, the correct answer is 4. So, look into this question also. Good question. Loss of water in C4 plants compared to C3 plants for the same amount of carbon dioxide fixed is. Always remember, friends, half. According to the transpiration ratio, suppose example, C3 plant lose 100 water molecules. C3 plant lose 100 water molecules to fix 1 glucose. Example. What about the C4 plant will do? C4 plant will lose only 50 water molecules to fix 1 glucose. So what will be the answer? Obviously, half. Not to get confused with that. Okay. In an experiment, it's a direct question. In an experiment, they are studying the photosynthesis. You provide a plant with a radioactive carbon dioxide as a metabolic tracer. They are saying that the 14 carbon dioxide, that is the isotope, is incorporated in the oxaloacetic acid. Here only you can understand the answer. Oxaloacetic acid only present where as a intermediate product in the C4 plant and CAM plant. Anyway, in the C3 plant, heterotroph is not the answer. Then you will get a confusion between the C4 plant and CAM plant. So you have to understand they performed during the day. You know, right? CAM pathway will occur during the night time, but they clearly given during the day high light intensity. Hence, the correct answer is what? Undoubtedly C4 plant. Very good question it is. Okay. Now, the basic feature of a typical C4 plant showing cranch anatomy. I have already told you now, friends, cranch anatomy, mesophyll cells are closer to the leaf surface, having lot of intercellular spaces. Bundle sheaths are located very deeper, no intercellular spaces. So, very good question it is. The presence of chloroplast in bundle sheath and epidermal, wrong. The presence of chloroplast in mesophyll and epidermal, that is also wrong. The presence of a typical chloroplast with well-formed granine bundle sheath cell, rudimentary chloroplast. Rudimentary means a kind of movable chloroplast. That is also wrong. Answer is, very good question it is, the presence of non-granule chloroplast in bundle sheath cell and a typical granule chloroplast in the mesophyll. That's why in the non-granule chloroplast only Rubisco is present. And granule chloroplast, PEP carboxylase is present in the mesophyll, not to get confused. Hence, the correct answer is 4. Which of the following statement is false in case of what plants, friends? In case of a C4 plants. Carbon dioxide acceptor is Rubisco in the mesophyll cell, which is a wrong statement, which is the PEP. So this is only wrong statement. Carboxylation occur in mesophyll, right? Leaves have two cell types, mesophyll bundle sheath. Mesophyll cells lack Rubisco. That is also right, because mesophylls are having what? PEP carboxylase. Hence, the wrong answer is what? Option 1. C4 plant can go on photosynthesizing even in lesser carbon dioxide. Friends, we discussed C4 plants are efficient plants in photosynthesis when compared to the C3 plant. What is the reason? The reason is very clear. C4 plants do not show photorespiration. So even though carbon dioxide level is very low, what is the enzyme? Every student remember, PEP carboxylase enzyme having higher affinity towards carbon dioxide. That's a very important point. PEP carboxylase having the lesser affinity towards the, higher affinity towards the carbon dioxide. So even though carbon dioxide level is low, it can easily fix it. Answer is 1. So, okay. In CAM plants, one second. In CAM plants, CO2 is taken up by dash through the stomata. It's a common, right? CO2 entered into the cell. How, friends? Via stomata. Because stomata is the one which is involved in the gaseous exchange. And where the leaves are present. So suppose this is the leaf we already studied. This will be the upper epidermis. This will be the lower epidermis. Lower epidermis contain guard cells. In between the guard cell only stomata is present. So where the leaf is present? On the green stem. On the green stem only leaf is present. Answer is what? Leaves on green stem. Simple question. In CAM pathway, first dicarboxylic acid is formed as a resultant of. I already told you. First of all, what is the first dicarboxylic acid? Oxaloacetic acid only. In case of CAM pathway also, PEP combined with carbon dioxide giving rise to oxaloacetic acid. But where? In the night time. In which cells? Of course, CAM plants also contain only mesophyll. So it also occur only during the night time. Not to get confused, friends. Carbon dioxide, NADPH, ATP ratio in the CAM plants. This is also a very good question. You have to compulsory remember this also. 1 carbon dioxide, 2 NADPH, and 5 ATP is required for 1 CAM cycle. Not to get confused, right? Hence, the correct answer is option 3. Okay, so friends, that ends all the questions only from the topic of photosynthesis. So in the topic of photosynthesis, I have solved 100 questions. Tomorrow, what we are doing tomorrow and day after tomorrow, I will solve 100 questions from cell organelles. And once, I mean to say tomorrow and day after tomorrow. Once again, we will come back to what? Once again, we will come back to the plant hormones and nitrogen metabolism. From these two topics, I will solve 150 MCQs. We will do 2 chapters simultaneously. Okay. Okay, friends. Thank you. Thank you, all of you. Thank you.